We've been spending a lot of time looking at the structure of our proteins. And throughout this, I've always emphasized that the structure relates to the functions of our proteins. So what do these proteins actually do? Well, again, of course, we can divide our proteins into two main classes. Our structural proteins, which of course play a structural role, also called fibrous proteins. And then we have our globular proteins. Globular proteins tend to be the more common proteins in our body, and they have a very wide range of different functions. We have proteins that store ions in molecules. We have proteins that transport ions in molecules, proteins that will defend against pathogens, proteins that are involved in muscle contraction and movement, and we have biological catalysts that will help to control the rates of the reactions within our body. Now, for many of these different protein functions, they require that our protein bind to a target molecule. So whether you're storing something or transporting something or catalyzing a reaction, in every case, you're going to have to interact with something else in order to carry out that specific function. Now, the binding target of our protein is normally called a ligand. Typically, this would be a small molecule, right? So you might be hemoglobin binding to oxygen, or you might be a enzyme binding to glucose in order to break it down to create energy. But it could also be larger molecules, right? So proteins do interact with each other, for example, or they also bind to DNA when we want to copy our DNA or convert it into RNA for producing new proteins. Whenever we bind to a ligand, so we're going to have a specific region within our protein that's responsible for that binding interaction. We refer to this as our binding site, right? So that region will have the particular amino acids that we need in order to form maybe a hydrophobic site or a site with negative charge to bind to something that's positively charged. Depending on what ligand we have, that binding site will look different in order to match with that ligand, right? So we're using the same kind of non-covalent interactions that we use to define our protein structure in order to interact with that ligand. One of the key features of this is that it allows the interaction to be transient, right? We don't want to interact forever. We don't want them permanently linked together. That would be more useful in a structural role. But when we are storing something, right, we want to be able to grab and hold it, but then release it when needed. Same thing for transportation or for catalyzing a reaction, right? You're always grabbing something and then releasing it at a later time. Because ligand binding is so important for our proteins, I want to spend a little bit of time looking at this from a quantitative perspective. So our protein-ligand binding interaction we can model as a chemical reaction, right? Essentially, we're taking one protein and we're binding our ligand to it and creating this new protein-ligand complex. So this creates, or this has an equilibrium, just like any other chemical process, where we'll have a balance between our bound form with the protein-ligand complex and our free form with the protein-ligand each free in solution. So since we have an equilibrium here, we can describe it based on an association rate constant, right, telling us the rate at which our ligand binds to our enzyme or our protein. And then we have a dissociation rate constant that refers to the rate at which our complex dissociates to get our free protein and ligand. Now, of course, if we remember from general chemistry, the rates of our reaction depend on our rate constant. So that's the Ka and the Kd for our forward and reverse reactions, multiplied by the concentrations, right? Concentration, of course, always influences our rate. So we have the concentration of free protein and ligand and the concentration of our protein-ligand complex. At equilibrium, these rates are going to be equal, right? So we have equal amounts of our protein-ligand complex forming and breaking apart, right? That's the definition of any equilibrium. The rates of your reactions have equalized and your concentrations are no longer changing over time. This, of course, also gives us our equilibrium expression, right? So as usual, our equilibrium constant is equal to our products over our reactants, each raised to their stoichiometric coefficients. So we have our protein-ligand complex as our product over our free protein and ligand. We can also reverse this to create a dissociation equilibrium constant, which would just be the inverse of our Ka, our association constant. Now, you should notice from these two equations that our equilibrium constant, of course, is related to our rate constants. So if we divide both sides by the free protein ligand and by the Kd, you see that the rate constant is actually equal to the ratio, or the equilibrium constant is equal to the ratio of our rate constants. So Ka over Kd equals Ka equals our ratio of products over reactants. Again, that's just showing that the equilibrium constant really depends on our two reaction rates in forward and inverse direction, right? Again, equilibrium is arrived when those rates have equalized. Now, in many cases, it's going to be difficult for us to measure the concentration of free protein and free ligand at any given time. So it's usually easier for us to refer to our amount of bound protein as the fraction of occupied binding sites. And we use the Greek letter theta here to represent this. So that fraction of occupied binding sites would be represented by the amount of protein that has ligand bound, so the protein-ligand complex concentration, divided by the total protein concentration. So this would be the concentration of our complex plus the concentration of free protein. Again, these can be difficult to measure by themselves, but we're usually always going to know what the total protein concentration is, right? If you imagine you're carrying out an experiment, you'd be adding some amount of protein, and so you would know, okay, I added 5 grams of protein, and I know the molecular weight, so I can determine the exact concentration of my protein. So as long as I have some means of measuring how much of my protein has ligand bound, I can calculate this fraction of occupied binding sites. Now, if I take these two equations, I can combine them together to get a relationship between my bound fraction and my Ka. So I can combine those like this. I'm just taking my protein-ligand complex concentration and substituting that using my equation here, right? So the protein-ligand complex is equal to Ka times free protein concentration times free ligand concentration, as defined by my equilibrium expression. And so if I just stick this value in within my equation, I get the expression down here in the lower left. Now, this expression isn't particularly useful, but you can notice here that I can now cancel out my free protein concentration, right? If I divide the top and bottom by the concentration of free protein, those will cancel out. And if I then divide by Ka, I would get this expression here. So I cancel out the ligand concentration, or the Ka on the top to leave just the ligand concentration. I cancel it on the left, and that leaves 1 over Ka on the bottom right. 1 over Ka, of course, is also equal to our Kd. The Kd and the Ka are the inverse of each other. And so I can express my fraction of bound protein sites as the ligand concentration divided by the ligand concentration plus this Kd, the equilibrium constant of the dissociation of my ligand from the protein. Now, this equation here is quite useful, right? What it tells us is that as long as we know the concentration of free ligand and the equilibrium expression for our, or the equilibrium constant for our protein, we can actually calculate how much of our protein will have ligand bound and at any given time, right? So we can actually remove the dependence on our protein concentration, essentially. So what would this look like in graphical form? So here we have a graph showing, this is the bound protein fraction on the y-axis, and our ligand concentration. And we're always going to see kind of this sort of typical curve. As we increase our ligand concentration, our protein is going to bind more of that ligand, and then it's going to slowly saturate over time, right? Because all of our, or almost all of our protein will have ligand bound, and it becomes harder and harder to get that final last few proteins to bind ligand to actually reach all of our protein having ligand bound. Now, one of the important assumptions that we usually make here is that our ligand concentration, our free ligand concentration, is usually going to be close to our total ligand concentration, right? So free ligand concentration can also be difficult to measure, right? You have to have some way of differentiating between how much ligand you have in solution versus how much is bound to your protein. But usually we have a lot more of our ligand than we do of our protein. So even if some of our ligand is bound to the protein, we can say, okay, it's still close to the same amount as our total ligand. So at this point now, as long as we know how much ligand we've added, right, what our total ligand is, we can determine how much is bound to our protein. Again, that bound protein fraction there. So at this point, from our graph, we can also easily determine the KD from our bound protein fraction. So the KD is actually going to be right here when we reach our half of our protein bound to ligand. Our KD is the ligand concentration. So this actually comes directly from our equation here, right? So if you substitute 0.5 in here for our bound protein fraction, you can then see that this would simplify to 0.5 ligand plus 0.5 KD equals our ligand concentration. If we subtract our ligand concentration from both sides and divide by 0.5, this simplifies to KD equals our ligand concentration. So again, you can see that essentially relates to what concentration of ligand we need in order to bind half of our ligand to our protein, or half of our protein bound to ligand, rather. Again, we're assuming that our ligand is much higher than our protein concentration, so the amount of ligand binding to our protein is relatively small compared to the amount of total ligand that we have. So we see then that this sort of graph has multiple benefits here, right? We can use to determine how much ligand will be bound to protein at any given time, just based on our ligand concentration. If we don't know what our KD is, we can also use it to determine our KD, right? As long as we know what fraction of protein has ligand bound, we can just measure the ligand concentration where we have 50% bound, and that would give us our KD. So you can even compare this for two different proteins. So here we see protein A and protein B. You can see their binding curves over time as we increase our ligand concentration. And from a quick glance at this graph, right, we know that at 0.5 is where we get our KD. So A would have a KD of around 2 micromolar, and B would have a KD around 6 micromolar. Now notice here that A binds, more of our binding sites are occupied at lower concentrations. So we would say that A has higher affinity, and B has lower affinity. So in essence, right, a smaller KD leads to a higher affinity. And this makes sense, of course, because our KD is that equilibrium constant that tells us how much of our protein-ligand complex is disassociating to our free protein and ligand, right? So a smaller equilibrium constant would mean that less of our protein is disassociating, or more of our protein is binding that ligand and forming our bound protein complex here. Now, of course, as usual, we can also relate these equilibrium constants to the free energy of this binding process. So we have our equilibrium expressions for the association and the dissociation, and these will relate to the free energy of this binding process as well, right? So in previous lectures, we've looked at the free energy of different different interactions, whether it's the enthalpy of forming new interactions or the entropy of changing the order of that environment there. And these will, of course, relate directly to our equilibrium constants here. So as usual, the free energy relates to the equilibrium constant through a natural log multiplied by the ideal gas constant and the temperature. So again, we can use those sorts of theoretical free energy calculations to relate directly to this equilibrium constant that will determine how much of our protein is actually binding to that ligand. So this is, of course, showing our expression for the association constant. We can also substitute in our dissociation constant, our Kd, and that would just change the sign here for our free energy. Again, it's just the inverse of our Ka. So our Kd, right, will relate to the relative amount of protein that will bind to ligand. It also will relate to the free energy of that interaction. And so the Kd is generally a convenient measurement for the overall strength of the interaction between our protein and our ligand. In general, a strong binding interaction, it would be around the nanomolar range. Again, of course, Kds are in concentration units. They also, of course, relate to the ligand concentration, so it uses the same units there. A weaker binding would be something that's in the high micromolar to millimolar range for our Kd. So we can actually look at varieties of different protein-ligand interactions. So you can see antibody and antigen interactions tend to be very high affinity. So we need to be able to detect very small amounts of invaders or pathogens in our environment. So our antibodies have to have very high affinity interactions to be able to sense and grab onto those. Enzyme to substrate, which is the term for an enzyme's ligand, these interactions tend to be much lower in affinity, right? Here we tend to have much higher concentrations of that substrate in solution. So we don't need as strong of an interaction as we do for our antibodies. You'll see receptor-ligand interactions tend to be ranging in affinity, but they tend to be somewhat stronger than enzyme-substrate interactions. So here you're often binding to a signaling molecule that might not be particularly high in concentration as well. One of the highest concentration, or one of the highest affinity interactions that we know is the interaction between biotin and avidin. So this is a protein-ligand interaction that actually approaches a covalent bond in terms of its strength of the interaction here. So it's somewhat unusual in that regard. So this interaction can be utilized in various experimental forms to link things together. It's also used in the body in some places as well. So how do our proteins achieve these high affinities, right? How do they target one specific ligand and make sure that they bind to that ligand in a very favorable interaction? So proteins will typically have very high specificity. They only will bind to certain ligands, right? Specificity just means that it's very specific for what molecule it binds to. The ways it does this is through complementarity in its size, shape, charge, and polarity, right? Again, similar to our protein structure, we're looking to make interactions between our protein and ligand that will stabilize that interaction there, right? So it's often useful to think of this as kind of a lock and key, where we have a particular shape, our lock, that will match the shape of our ligand so that it will fit right into our protein there. Now, while this lock and key model is useful for thinking about the complementarity that we have to have between our protein ligand, in many cases, our proteins actually use more of an induced fit model. So in an induced fit, we might have a shape that's initially somewhat complementary to our ligand. But as the ligand comes in and binds, we're going to change the shape of our protein. So we have a conformational change where it slightly adjusts its shape in order to increase that complementarity and bind to our ligand. So this has a number of different advantages. It can allow for higher affinity as we kind of adjust to make sure that interaction is as good as possible. It can also give us a little bit more flexibility, right? You can maybe adapt to different ligands if you need to bind to multiple different ligands. It also can allow us to change the strength of our interaction, right? So this helps with regulation. You might be able to tell a protein, oh, no, it's time to stop binding to this ligand, so I'm going to change my shape in order to release that ligand. Generally, conformational changes are very important for our ligand binding interactions. In order to gain a better understanding of our protein ligand binding interactions and protein functions in general, I want to take a very close look at a particular class of proteins. So these are globins, which are oxygen binding proteins. So globins use a very unique approach to solve a key biological problem. So of course, in our biological systems, we have a very strong need for oxygen. The problem is that our protein side chains of our common amino acids don't have a high affinity for oxygen, right? None of those different amino acids that we've looked at, lysine or glutamate, they don't bind to oxygen by themselves. If we want to bind to oxygen, usually we need more organometallic compounds, right? Things like iron can bind well to oxygen. Iron found inside of a heme group, which has that iron coordinated to some nitrogens inside of a organic and metallic structure. So this is more useful for binding to heme, but we can't just use it by itself in our body, right? So if you just have that heme sitting around with it uses iron with the 2+ charge, it can get oxidized to our iron 3+, that then becomes reactive and can cause other issues within our body. So we can't use just a protein, but we can't use just an organometallic compound. The solution then is to use a heme molecule, use our organometallic compound, but bind it inside of our protein, right? So we can use our protein as a capsule, essentially, to carry this heme group that will then carry our oxygen. So both myoglobin and hemoglobin use this protein-bound heme approach. Myoglobin uses it for storage of oxygen, and hemoglobin uses it for transport of oxygen around our body. So here's what our myoglobin protein looks like. So each of these cylinders here are representing alpha helices. So you can see we have mostly alpha helical structure. It is in a kind of compact globular protein shape. And this red right here is our heme group that's bound in the center of our myoglobin protein. So the orange is representing our iron here. That's going to be actually binding to our oxygen. And we have interactions that will occur between side groups of our protein's amino acids. You can see these histidines noted here, and our heme group to help to bind that within our protein. So as with any protein-ligand interaction, we can describe our myoglobin using our typical equation, right, relating the bound protein fraction to the ligand concentration and the KD. However, for myoglobin, we're dealing with a gas, right? So our ligand is oxygen gas, and so we have to rephrase our equation here in terms of partial pressures, right? Because our gas isn't dissolved in solution in the same way that another ligand might be. So rather than concentrations, we express this as partial pressures of oxygen. So the partial pressures essentially are concentration here, and instead of a Kd, we use this P50, which is just the partial pressure at which 50% of our protein has oxygen bound. So again, it essentially means the same thing as our Kd, we're just using a slightly different term here. Let's take a closer look at our heme structure here. So again, our heme is an organometallic compound, meaning that we have an organic component, shown here, as well as a metallic component in the middle. So our iron is going to use coordinate bonding to interact with lone pairs on these nitrogens. So we have 4 different nitrogens surrounding it in this complex ring system. So you can see we have this ring of carbons with our nitrogens all pointing in to grab onto that iron. You can see that noted here as well. You've got the gray of our carbon ring surrounding it, the blue of our nitrogens, and the orange of our iron right here in the middle. Our iron, of course, interacts through those coordinate bonds where it shares lone pairs with nearby atoms. If you remember from organic chemistry, our iron atom wants to gain 18 electrons according to the 18 electron rule that we learned for organometallic compounds. Iron 2+ initially has 6 valence electrons, and so we need to gain 12 more in order to have that 18 electron rule fulfilled. So you can see we're getting 8 here from our surrounding nitrogens. This is all kind of a planar ring structure. So we have one plane of our ring with our 4 nitrogens surrounding our iron, and then we're going to form 2 more interactions from above and below in order to get our full 18 electrons. So those last 2 coordinate bonds are formed to oxygen, our ligand, and to our protein, again, to hold our heme group within our protein. So you can see here, this is an edge view. So we're looking at the side of our ring. So our ring is perpendicular to the screen here. So here would be all of our nitrogens surrounding that iron and forming those coordinate bonds. And then we have a histidine nitrogen. We refer to this as the proximal histidine. It approaches from below and adds 1 more bond to our iron. And then the oxygen uses its lone pair to occupy the last binding site there. So you can see this in our protein structure on the right as well. Here's that proximal histidine. This would be histidine 93 in the amino acid sequence of our myoglobin. It's binding right here to our iron, and our oxygen would be sitting right here on the other side, interacting with that iron as well. So again, the advantage of this is that our iron is very nicely kind of surrounded and bound inside of our pocket and protected from any oxidizing agents that would react with that iron there. Now, the unfortunate thing is that our iron can also bind to other ligands other than our oxygen, right? So again, oxygen would be using this lone pair here in order to bind to our iron. It ends up binding with this bent shape because, of course, our iron itself is sp2 hybridized with a trigonal planar shape, right? So we have these lone pairs sticking out at essentially 120-degree angles in order to get our trigonal planar shape, and one is pointing directly down to bond to our iron there. Carbon monoxide, we have a carbon atom that also has a lone pair that can bond to our iron, right? So in terms of size here, carbon monoxide is quite similar with that just 2-atom gaseous structure. And our carbon, of course, is much more willing to give away electrons than our oxygen because of its lower electronegativity. So it can actually form an even stronger bond to our iron. So this is actually the cause of carbon monoxide poisoning. So it binds and replaces oxygen in our heme groups within our hemoglobin and myoglobin. So because of the lower electronegativity of our carbon, carbon monoxide actually binds roughly 20,000 times better than oxygen to our heme group. Now, within myoglobin, we can try to protect our oxygen and prevent carbon monoxide from kicking out that oxygen. So one of the things that we do to try to reduce the affinity for carbon monoxide is we bring in another histidine that we call the distal histidine to interact with our oxygen on the other side. So notice, of course, we have all of our coordinate bonds down here around our iron, to the nitrogens within the ring, as well as to that proximal histidine, and to our oxygen ligand. Now, the oxygen sits in this angled shape because of the sp2 trigonal planar nature of our oxygen, and our histidine here is perfectly placed to hydrogen bond to that oxygen with its extra lone pair. So it forms that interaction with our oxygen, whereas our carbon monoxide, since it would want to be sticking straight up and down, doesn't interact with that histidine. So the histidine kind of stabilizes our oxygen and helps to destabilize carbon monoxide. Now, unfortunately, this can't completely stop carbon monoxide, so this reduces the affinity, but carbon monoxide still has roughly a couple hundred times higher affinity for the iron than our oxygen. So it doesn't completely solve the problem, but at least it reduces the affinity there for carbon monoxide, and so it makes it less, it requires more carbon monoxide essentially to replace our oxygen and cause the negative health effects of carbon monoxide. Now you can notice here as well, we do have other amino acids that are going to be around our heme group as well. So we have a valine and a phenylalanine noted here. Well, these will help to stabilize our heme structure, so they'll be interacting with the other parts of our heme structure to help to hold that within our protein, right? So the heme is not covalently bound in our protein. It's just kind of surrounded and kind of packed in this binding site. So you could theoretically lose your heme group from the protein if you didn't have these amino acids surrounding it and holding it in place. Our myoglobin here also provides a great example for showing how we can measure the bound protein fraction for a given protein-ligand interaction. So within our myoglobin, again, we have that heme group that's binding to our oxygen. Oxygen would be our ligand. And the heme group has an iron that actually changes its absorption as it binds to oxygen. So in the ferrous form without oxygen, we absorb at 429 nanometers, whereas once we bind to oxygen, it actually alters the electronic properties of our heme group, and our absorption shifts to 414 nanometers. So because of this, our deoxyhemoglobin or myoglobin appears more purple, and our oxyhemoglobin or oxymyoglobin will appear more red. This is actually one of the key things that causes the difference in color between our veins and our arteries. It's actually just whether our hemoglobin that's carrying the oxygen has oxygen present or whether it's already released the oxygen, right? So it's carrying oxygen out and then it's coming back mostly devoid of oxygen, and its color changes during that process. So we can see this in graph form here. So this graph is just showing, so you can see this peak here from our deoxy when our ligand is not bound, and we have another peak from our oxyhemoglobin or myoglobin when we have our oxygen bound. So if we just tracked the percentage of absorbance at each of these wavelengths, we'd use that to determine the relative fraction of our protein that has the oxygen bound. Right, so again, we're just looking at when our oxygen is present, we have the extra interaction with our heme group. When it goes away, our interaction is gone, and it kind of shifts the position of our heme and then also ends up shifting that wavelength there. And this effect is also seen for other proteins as well. So there are proteins that can be used as oxygen sensors that will have that same kind of shift when oxygen is present or not, and you can measure that again just through absorption. So if we measure this for myoglobin, so we would again be able to use absorbance to figure out a bound protein fraction. And if we use different concentrations, or of course in this case, partial pressures of oxygen, we can see exactly where the P50 for myoglobin would be. Now, one of the things that you might notice here with myoglobin is that it has a very high affinity for oxygen, right? So it doesn't take a very high pressure of oxygen for us to reach 50% saturation with that oxygen bound to our protein. And as you increase that concentration, we're going to quickly increase until we hit close to 100% of our myoglobin bound with that oxygen. So if we look at our lung partial pressure of oxygen, it's roughly 13 kilopascals. So that results in roughly 98% of our protein having oxygen bound, right? We'd be up here above the top of our curve. We're hitting almost that 100%, but just slightly below. In our tissues where we need our oxygen, the partial pressure is about 4 kilopascals. That would be somewhere around here on our chart. And you can see we still have a very high degree of oxygen bound to our protein. There's this very high degree of oxygen saturation. So if you calculate it out, it comes out to roughly 93%. So between the lungs and our tissues, if myoglobin was moving around trying to transport oxygen, we would only have a roughly 5% release of oxygen. So oxygen is very inefficient at transporting, or myoglobin is very inefficient at transporting oxygen because it binds to it too strongly. So that's why myoglobin is more of a storage protein, and we need a separate protein for transporting our oxygen. Now, this is kind of a difficult problem here, right? Because that sort of heme binding has a very high affinity for oxygen. So we need some way of actually switching from a state where we bind oxygen too tightly and won't release oxygen to a lower affinity state where we're able to release oxygen more easily. So you can see this line in the middle is representing a protein that's able to transition between high and low affinities in order to make sure that it's binding lots of oxygen in the lungs, but then releasing lots of that oxygen once it gets to the target tissues. So our protein that accomplishes this, of course, is hemoglobin. It's going to use conformational changes in order to change the affinity of the ligand for the protein. And we'll take a closer look at how that works in a separate lecture.